by way of introduction, yeah, this is my office. No, I'm just kidding. And probably I'll retire over there. At least that's the plan. We'll see what happens. By way of introduction, let me just say that this is a sentence that you often see in biomedical literature, in recent literature, but also some time ago. These and other findings suggest that protein X may offer an effective way to target some malignancies. I'm just trying to explain how I entered into this field of research into single domain antibodies. So this is a sentence that still today you see in many different papers. And so you get the impression that each and every protein at some point is an interesting potential target. However, there is a kind of problem because many proteins are considered to be undruggable. And by that, I mean structural proteins, scaffolding proteins, non-enzymatic proteins. But according to statistics, over 80% of disease-related pathogenic proteins cannot be targeted by current methods. And by current methods, I mean in a pharmacological way. And undruggable, so it means that proteins cannot be drugged by small pharmacological compounds. And a long time ago, I thought that maybe single domain antibodies might be a way of addressing this issue. I was also inspired to some extent by Sidney Brenner. I hope you know him, you should know him, because he said progress in science depends to a very large extent on novel technologies. And back then, single domain antibodies were a relatively novel technology. Very brief history. Where and when were, I'm going to use this term, VHH, single domain antibody, nanobody, it's all the same thing. They were discovered already a long time ago in an immunology lab at the Free University of Brussels in Belgium. And actually, sometimes we joke it was a typical Belgian discovery. Nobody was looking for them, but the undergraduate students kind of tripped over them by accident. They discovered them in a practical course, not the professors themselves, of course. Professor Geltemans is still around. He's a very nice person, and he was actually one of the persons involved in the discovery. The first paper published in '93 already. You can see the conventional heavy and light chain of normal IgGs. And next to that, you see a single band. You see a heavy chain, but you don't see the light chain. And the rest is history. So I probably don't have to explain to this audience what a conventional antibody looks like. You have the heavy and the light chains. The camelids, so the llamas, alpacas, the animals that have these heavy chain antibodies, they also have regular IgGs. But they're endowed with a second type of antibody called a heavy chain only antibody, which lacks light chains for the simple reason that the CH1 domain is lacking. The CH1 domain, constant heavy, first one, that's where the light chain is anchored to. If that domain is missing, you cannot anchor a light chain onto a heavy chain. So basically, a VHH or a nanobody corresponds with the antigen binding parts of such a heavy chain antibody. And it's 10 times smaller than an IgG, 15 kilodaltons. These are the animals where you find these heavy chain antibodies. Some of them are not listed. A nurse shark, a wobbegong shark, they have similar, it's not entirely identical, but very similar to these heavy chain antibodies as well. The key properties, let me briefly summarize why I think these molecules, these antibody fragments might be interesting for research, therapeutics, and diagnostics as well. First of all is you always have the DNA. You have the DNA by default, and because of the fact that nanobodies do not require PTMs, no glycosylation, no post-translational modifications, which means you can easily express them in E. coli or in yeasts. And because of the fact that you have the cDNA, you can also relatively easy derivatize them. I'll give an example further down in the presentation. So you can tailor, you can manipulate, you can change the DNA. And by doing so, you can change the properties of the single domain antibody. Production at very low cost, no mammalian cells, bacteria suffice. They are actually monovalent, monoclonal antibodies. And even in a monovalent format, they bind with very high affinity. Equilibrium dissociation constant 10-9 up to 10-15, which is very strong, 1 femtomolar. Small, 15 kilodaltons, a single chain of amino acids, 120 of them, and at the DNA level, only 360 nucleotides. And in principle, you can modulate any type of protein in vitro and in vivo. And again, I'll try to give a few examples. I'll give the presentation mainly from a technological point of view. I'm not going to discuss everything that has been done. I'm not going to give a historical overview. I'm just going to try to present some case studies to illustrate where they might be of additional value in comparison to conventional antibodies. Well, one slide comparison, we can skip this. I just mentioned the most important properties that are relevant for this discussion. It took a very long time, but since a couple of years, a number of years, nanobody-based drugs are available. The first one was approved in 2019 by FDA, was developed by Ablings, incidentally a company on the campus where I work now. But in the meantime, it's been taken over by Sanofi, big pharmaceutical company. And Caplivi, as it is called, was developed by Ablings to treat a kind of rare disease. It had something to do with blood clotting, which is a very complex mechanism. Many different proteins are involved in the clotting mechanism. To cut a long story short, the nanobody targets von Willebrand factor. And what it does is it prevents so this one here is Caplivi. It's actually 2 copies of a nanobody. And the polymers that you see there is von Willebrand factor. And what the nanobody does is prevent interaction between thrombocytes and von Willebrand factor, so that you don't have microthrombi, which apparently are very dangerous for those patients. They have a certain disease in which ADMTS13, some enzyme is lacking. And the enzyme cleaves von Willebrand polymers into small fragments. Another one is called, another drug, Nanosora, is approved in Japan for treatment of arthritis. And this already shows an interesting property of nanobodies is that you can concatenate them. You can use 1 copy, you can use 2 copies that target the same protein, or another nanobody that targets another protein. In the case of Nanosora, you have 2 identical nanobodies against TNF, tumor necrosis factor alpha, and these 2 are coupled to another nanobody that binds with albumin, so that the circulation time increases, because otherwise it's a bit too small and is going to be cleared from the circulation rather quickly. So these are 2 examples. No doubt other companies are making new nanobody drugs for breast cancer, arthritis, myasthenia gravis. It's a neuromuscular disease, Crohn's disease. Anyway, these are being developed by other companies. You might wonder, how do you make them? How do you obtain a set of nanobodies against a given antigen? Well, one way to do this, because there are several ways of approaching this problem, but one way to do this is to purify an antigen. Any protein is fine, it shouldn't be too small. Peptides, for some reason, are not good antigens for raising nanobodies because they're basically too small. Nanobodies like to recognize extensive conformational epitopes, which are lacking in small peptides. So this is a big protein you inject. We don't have camels, we have llamas. They're already big enough and they eat a lot of food. We immunize llamas. 6 weeks, after 6 weeks, we obtain a small quantity of blood, isolate lymphocytes. From the lymphocytes, we extract messenger RNA, and by RT-PCR and 2 sequential PCR reactions, we amplify by PCR the entire repertoire of each and every nanobody that is present in the lymphocytes, not only against your target of interest. So we obtain a huge library of VHHs, or single domain antibodies, here. And then we clone them into a phage mid. A phage mid is basically a plasmid, but it can also be transformed into a phage, a bacteriophage. So we electroporate the entire library of the VHHs into coli, a certain strain, TG1. And then afterwards, we add a helper phage. And then this is going to result in a huge collection of bacteriophages, starting from the phage mid. Yeah? So you make a collection of bacteriophages with each phage having one single nanobody on its surface. And then by phage panning, we are here now, by phage panning, which means basically it's a kind of ELISA technique. You spot the antigen on the surface of a 96 well plate. You add the entire collection of bacteriophages. The ones that express a nanobody against your target of interest will be retained. All the others are washed away. You repeat that procedure a number of times, and by doing that, you enrich for specific binders against your antigen of interest that you injected in the first place. So the last step is, this is not how to pull out the nanobodies from the animal. I just explained that you do this by phage panning. So what is the typical end result of such an immunization program? You end up with a number of nanobodies. You immediately have the sequence because you have the DNA. So you translate the DNA sequence into the amino acid sequence. And what we look at, I think this was a project against it for BCL2, mitochondrial protein or a mitochondrial associated protein, if I remember correctly. What is interesting is you always have multiple nanobodies, not just one. It depends on the antigen and it depends on the immune response of the animal. 2, 5, 20, it depends. In this case, we had like six different nanobodies. And you know, they fold up like a typical antibody. You have constant framework regions, you have variable CDRs or complementarity determining regions. And we are usually very interested in looking at this region. The higher the diversity of CDR3, the more different nanobodies you have. You can group them into different families. And potentially, you're not quite certain about this, but potentially they might interact with different epitopes. So you can use these different nanobodies to screen the surface of your antigen, looking for interesting epitopes maybe, or maybe some biological effect that you can block using that particular nanobody. What opportunities do they offer? Well, I always make this comparison with a planetary rover that scans the planet for interesting topical features on the surface. We use nanobodies for scanning the surface of the antigen. For example, if this would be the antigen that you injected, you end up with a family of different nanobodies. One will bind to this epitope. Apparently, they don't have any biological effect. They might be interesting as a negative control. Other nanobodies bind to another region. They can block interactions between proteins. In other cases, they can block the enzymatic activity of your protein of interest. And what I'll try to show in the next slides is that indeed they can be used to drug undruggable proteins. We're going to skip this one, except for the fact that I would like to say the following, that CDR3 in these antibodies is usually longer than the CDR3 in human or mouse antibodies, which in part explains why these long CDR3s are able to bind into a catalytic clefts of an enzyme and block the biological activity, something which is a little bit more difficult to achieve with an IgG because it's kind of bulky. What is also of interest to know is that overall, the primary structure of a Camelid antibody is much more similar to the sequence of human antibodies in comparison to mouse human. So you would expect that they are less immunogenic than mouse IgGs. I'm not saying that they are completely not immunogenic, but they are probably less immunogenic. And another interesting feature is that Camelids are outbred animals, which means they are genetically totally different one from the other. And if you inject two different llamas with the same antigen, it's always going to yield a totally different set of nanobodies. Never the same, never. Because they're outbred, genetically diverse. Let's take a moment and reflect on the difference between using antibodies versus CRISPR or genetic approaches. Maybe some of you have been thinking, why spend so much work in developing antibodies if you can do some experiments very quickly by genetic approaches, you know, gene expression modulation, CRISPR-Cas9, and things like that. I think there are some interesting differences to mention. In the case of gene regulation, the target protein expression is reduced or completely eradicated. You can no longer visualize the target because it's gone. You probably also disrupt protein-protein interactions. And something we tend to forget, finally, is that proteins have multiple functions, and all of them are obliterated when you use a genetic approach. By contrast, in the case of antibodies or nanobodies or whatever you want to use to target a protein directly, this is exactly what you do. You target the resident protein directly. You do not in any way affect the expression level. You can still visualize the target at all times. You can discover, this is the most important word, you can discover which protein-protein functions have been disturbed by an antibody or a nanobody, and you can also discover which target protein function is disturbed. So we should be a little bit careful if you use only genetic approaches and predict whether a protein is of therapeutic interest or not. I think it's always a good idea to have at least one complementary technology that is going to tell you whether your hypothesis was correct or not. Let's take a look at some of the advantages, and I'm going to focus specifically on what I see as an advantage specific to a VHH and something that is more difficult, I'm not saying impossible, but sometimes more difficult to accomplish with an IgG. I mentioned already concatenation. I'm going to skip this. I gave the example of nanosora, yeah, concatenation. There are quite some opportunities in diagnostics. You know, basically it's an antibody, it's a high affinity binder, and there are different possibilities in diagnostics as well. I'm not saying that antibodies or nanobodies better are used on a large scale in diagnostics because the market is still kind of dominated by IgGs, but I do believe that in some cases nanobodies might have interesting advantages. For example, as I'm going to show in one of the next slides, it is possible to derivatize a nanobody in such a way that you can orient it in a specific fashion onto a support. If each and every nanobody is oriented in exactly the same fashion, it is accessible to participate in the reaction with an antigen. Whereas in some cases, if you use antibodies, IgGs, and you do not orient them specifically onto a support, some of the antibody is going to absorb to the surface, it's going to denature, it's going to have the wrong conformation, or the epitope, the paratope is not going to be accessible for interaction with the antigen. By the way, it might also be possible to directly couple a reporter enzyme, such as alkaline phosphatase or a peroxidase. You can make a fusion protein. And in that case, you don't need a secondary antibody anymore, because the nanobody that detects the antigen is immediately coupled to the reporter enzyme. And we just tested this very briefly. We made such a fusion protein and expressed it in mammalian cells, and this seemed to work very efficiently. So in that case, you don't need the secondary antibody anymore. But I have to admit, to my knowledge, I am not aware of the fact that these days diagnostic tests that are based on this principle are already available. I'm just trying to give some opportunities. Biosensors, very sensitive detection of antigens using electrochemistry. A while ago, we had a student from the sensor lab headed by Professor Iwoa at the University of the Western Cape. Jamie spent some time in our lab and she developed a nanobody-based biosensor to detect MCP, macrophage capping protein, a cancer biomarker. So she used two nanobodies that interact with non-overlapping epitopes of the antigen, one of which had to be biotinylated. One nanobody had to be biotinylated, but that was not too problematic because, again, keep in mind, you have the cDNA, you can modify the cDNA, and in this case, you can modify it by adding at the C-terminus a consensus sequence that causes biotinylation, that promotes biotinylation. If you put the plasmid into the bacterium, and the bacterium reads the consensus sequence, it's going to add a biotin group. So Jamie's work was quite successful because in the end, she didn't spend so much time in the lab, but she was able to generate a very nice paper that was published at the end of last year. And to be honest, I think there is still quite a lot of margin of improvement in this particular biosensor. Another application area for nanobodies is in nuclear medicine, using them as a tracer. You could argue we have antibodies that are useful as a tracer. I agree, but in some cases, these IgGs, they circulate for a long time. And the longer they circulate, the higher the background is going to be, the lower the contrast. Now, nanobodies are much smaller, which in many cases is perceived as a disadvantage because they're quickly cleared through the kidneys. But I think in this case, it can be an advantage. It's quite possible, and this has been shown in literature by many different groups, it's not too difficult to couple a broad variety of radionuclides onto a nanobody. For example, the TC technetium-99 is very easily coupled onto a nanobody through its histidine tag. Many nanobodies are expressed with a histidine tag to facilitate purification, and the technetium specifically interacts with the histidine tag. Because they're small, they will produce a lower background. And as a result of that, they will, in some cases at least, give a better contrast. And we've used a nanobody in one study, as I mentioned. We use here technetium-99, and it's a nanobody that recognizes amyloid fibrils. It's a disease that has some similarities with Alzheimer's disease, but the protein that is being cleaved, it's not APD. It's a different protein. I'm going to explain a little bit more in a couple of slides, but the nanobody recognizes amyloid fibrils, and this is what you can see in the preclinical model. This mouse develops the amyloid disease in a similar way to humans. Let's see if we can, just a second, yep. You have to focus particularly on the blue-purple regions. This is exactly where the amyloid deposits are to be found, because actually what you image there, in part at least, is skeletal muscle. And it's surrounding the myofibers that the amyloid fibrils can be detected. So in a non-invasive manner, you can use nanobodies also as a tracer in nuclear medicine. Maybe a little bit less relevant to what your research involves, I don't know exactly, but I would like to mention this at least, that one of the very distinctive features of nanobodies is that you can use them as intrabodies. Normally, antibodies are unstable in the cytoplasm. Are there many students in the auditorium? Can I address the question? Why antibodies usually are not stable in the cytoplasm? Keep in mind the light chains and the heavy chains. There's something that connects them, disulfide bridges, and you need the disulfide bridges for the antibody to be functional. But what happens in the cytoplasm? You know, usually antibodies never end up in the cytoplasm. They're being secreted. But if you purposefully put them in the cytoplasm, something's going to happen. There's a lot of glutathione in the cytoplasm in millimolar concentrations, if I'm not mistaken. Glutathione is a peptide of 3 amino acids, and the last one is cysteine. So if you have a lot of cysteine present in the cytoplasm of cells, it's going to reduce the disulfide bridges. Light chains and heavy chains each go their own way, and the antibody is usually not functional anymore. Nanobodies also have an intramolecular disulfide bridge, but for some reason it resists the high glutathione concentrations. And this gives you the possibility of doing a couple of different experiments. You can directly modulate, in principle, any target in the cytoplasm or in any compartment for that matter. You can also promote delocalization. You can take any protein and put it somewhere else in the cell. For instance, you could target a transcription factor that causes a disease, and you pull it out of the nucleus and put it somewhere in the cytoplasm, hypothetically. And it also allows you to trigger proteasomal degradation, similar to RNAi. This is one example. We're looking at one protein. We're looking at one cell, sorry. This is one cell. You can see a red color, the antigen. The protein is called CAPG, and it's present in the nucleus and in the cytoplasm, everywhere. However, if you transfect the cDNA that encodes a nanobody against this protein, and you add a subcellular localization sequence, this one here, MOM, stands for mitochondrial outer membrane sequence. It's just a small sequence that you tag onto the nanobody, and you will see that you get a reticular staining pattern. It's localized on the outside of mitochondria. And if you look at the staining pattern of the antigen, you see that it changes. It's completely identical to the nanobody. So it allows you to change the localization of any protein, and at the same time, it tells you that the nanobody is functional, because otherwise there wouldn't be any colocalization. You can put the same protein in the cytoplasm using a different targeting sequence, or you can put it inside the nucleus using a nuclear localization sequence. So that's a very distinctive feature of nanobodies, and there are, in the meantime, quite a few papers reporting interesting results using intrabodies to modulate a certain protein. And actually, this nanobody that is shown there has a very simple biological activity. CAPG is a protein of the cytoskeleton. We have in each cell approximately 140 different proteins that regulate the polymerization status of actin, which is important for migration, motility, and so on. CAPG is one of those 140. It interacts with actin, but this nanobody blocks the interaction between the two proteins. And this has some interesting properties. I'll tell you why in this slide. CAPG has been reported to be overexpressed in breast cancer, like so many other genes. So because we had this nanobody, we thought, suppose we transfect, or transduce, I should say, because we use lentivirus, suppose we transduce it into human breast cancer cells, and we inject the breast cancer cells in mice, female mice, orthotopic injection. What is going to happen? Well, we generated a stable cell line of MDA-MB231 cells that stably but inducibly express the nanobody under control of doxycycline. The absence of doxycycline, the nanobody is not expressed if you add doxycycline. And the expression level was approximately at the same level of the antigen. It was not a 10-fold overexpression. So we then injected those cells, but this is the control group. They do not have the nanobody. This is just injection of human breast cancer cells in mice. Non-invasive imaging, EVIS imaging, and you can see the purple blobs. This is where the cells were injected, and you see what happens over time. 38 days, and so on. At a certain point, for the students, sorry, metastases to the lungs. So at that point, you have to sacrifice the animals, otherwise the burden is going to become too high. This is exactly what you expect for human breast cancer cells. However, if we inject the same cells and they express a nanobody against CAPG, then you see something different. You can see the tumor still growing, but there's no metastasis. And actually, yeah, there was a reduction of 97% in photon flux coming from the lungs. So it has a very potent activity in terms of blocking cancer metastasis, at least in a preclinical model. And the point I'm trying to make actually is the following. I'm quite convinced that in the cytoplasm, there are many interesting therapeutic targets as well. And maybe we shouldn't always think about surface receptors, receptor tyrosine kinases, ion channels, and things like that. I think that within the cytoplasm, there are also many different potentially interesting therapeutic targets. And maybe because of the fact that they are at the bottom and not at the top of a signal transduction pathway, targeting them might trigger less side effects. But that's a hypothesis. I'm going to skip this one. The next example goes a little bit further. Here I want to show how you can use nanobodies in studying a disease that is caused by protein misfolding. It's an orphan disease. It's called familial amyloidosis of the Finnish type. Orphan disease, I'll explain immediately what I mean by an orphan disease. It's a monogenic disease caused by a mutation in gelsolin, and the gene encoding gelsolin. Now, just like CAPG, gelsolin is a textbook example of a cytoskeletal protein. And there is some similarity with Alzheimer's disease because it's an amyloidogenic disease, but the protein that is being cleaved is not APP, it's gelsolin. The mutation D187N means that an aspartic acid has changed into an asparagine. Aspartic acid is important in calcium coordination, and gelsolin is a calcium binding protein. However, when this residue is mutated, that particular domain is no longer able to bind calcium because the aspartic acid has been mutated. And when this happens, there is a kind of gain of function. Suddenly furin, it's an enzyme in the trans-Golgi network, suddenly furin cleaves off a fragment of gelsolin. And when that happens, there's another protease, MT1, a metalloprotease, and that one cleaves off amyloidogenic peptides, and they assemble into fibrils. Some of the clinical features are shown here. Here you can see amyloid fibrils in the eye, atrophy of the tongue, and this here is known as cryptisplaxa. I'm not saying it's a life-threatening disease. You're not going to die from it, but it's extremely uncomfortable. And even surgery in some cases has been tried, but it doesn't help very much. Gelsolin amyloidosis is an orphan disease. An orphan disease is a condition that affects less than 200,000 people worldwide. We all know several examples of orphan diseases: cystic fibrosis, Tourette's syndrome, Kuru disease, and so on. Actually, there are between 5,000 and 8,000 orphan diseases that still cannot be cured. So MDs and researchers in biomedicine still have a lot of work to do to solve, or at least to come up with a relevant therapy for these orphan diseases. Oh my goodness. He was also diagnosed with an orphan disease. It's probably a variation in Tourette. He received treatment and feels much better these days. I know what you're thinking. Well, I was not planning on going to the US anytime soon. Doesn't matter. To know what we can do about this disease, because these patients, what kind of treatment do they receive? Eye drops. Eye drops. It's just symptomatic treatment. It doesn't help in any way. We need to look at the molecular details of the problem, and we need to look at the trans-Golgi network, because that is where furin becomes active. So the red dot is the mutation in the protein, ER trans-Golgi network, and the yellow here is furin. Probably what you see happening is the following: a fragment of gelsolin has been cleaved off. Yeah. And at the moment of secretion, the second enzyme is waiting, MT1, and that one cleaves off peptides. It's shown here. Second enzyme, MT1, removes 70 amino acid peptide, and this one assembles into fibrils. Again, a question for the students. You know more or less the problem. What is your first target to, develop inhibitors against? If you want to block the system, well, you block one of the enzymes. That's the easiest way to approach it. Either furin, and actually inhibitors already exist, but they're not being used for treating this disease. And the same applies to MT1 MMP. Why don't we use them? The reason is not too difficult. It's a chronic disease. This is happening on a daily basis, 24/7, which means that you constantly have to provide these patients or treat them with inhibitors against furin or MT1. And that is not a good idea. Simply for the reason that furin, for example, to give just one example, it's necessary for maturation of neuropeptides. If you block that, I don't know the consequences. I don't know what's going to happen. So you cannot do that. It's going to cause numerous and very significant side effects. So what is left, at least that's an approach that we tried. I'm not saying it's the only one. We tried to do the following. Use the nanobody against gelsolin as a molecular chaperone, shielding it from proteolysis by furin or MT1. No doubts, other possibilities exist as well, but this is the approach that we tried. How did we do that? I'm going to save you the details. How we generated those nanobodies. They are available. We generated them years ago. Number 11 binds to the second domain, which is the domain where the mutation occurs. Then there's another one that binds to domains 4 and 5, and we use that as a control. That's number 13. So the first thing that we tried to do, well, we did first biochemical experiments, in vitro experiments. They were pointing in the right direction. And the next thing to do was, can we use that nanobody in living cells to protect gelsolin against furin proteolysis? We transfected hex cells. Hex cells contain furin. You don't have to transfect furin. They have the enzyme. We transfected mutant gelsolin in green, and we transfected the nanobody 11. However, gelsolin is secreted from the cells. So it's going through the ER, which means that you also have to equip the nanobody with an ER targeting sequence. Otherwise, they're never going to meet. So that's what I mean by ER, an ER targeting sequence, cDNA of the nanobody, and then some tags for visualization. Transfection of this nanobody, and there is a nice colocalization in these cells, meaning that they both colocalize in the ER. On their way out, they're being secreted. And you can see indeed in the medium that gelsolin is being secreted, of course, because it has a secretion signal. This is a Western blot. The upper band is full length, uncleaved mutant gelsolin. And the band below is the fragment that originates by furin cleavage. It's 68 kilodaltons. And in the case of nanobody 11, you see that there is less of the C68. So somehow, we don't know exactly how, but somehow nanobody 11 protects gelsolin against cleavage by furin. Whereas the one that binds, here in domains 4 and 5. Yeah, that one has no protective effect whatsoever. I'm not showing the Western blots, but it has nothing to do with differences in expression level. Both nanobodies were expressed at the same level. So we had one nanobody that protects against furin. I'm trying to summarize as briefly as possible. Later, we also identified a nanobody that protects against the second cleavage, MT1 MMP. And we combined both nanobodies into one cassette. The cassette of an AAV, an adenovirus associated, an adeno-associated virus, sorry. So what we generated here is a biparatopic nanobody, two different nanobodies binding to different sites in the target. Both have a protective effect. And then this virus, because basically it's, within the genome of a virus, we produced it at very high titer, and then it was injected behind the eyes of a newborn mouse. And the mouse is again the transgenic mouse that recapitulates the proteolytic cascade as observed in human patients. And this is the end result. This is a control staining to show that you're not cheating with the microscope. In the untreated animals, you see a lot of red surrounding the myofibers. This is exactly where the amyloidogenic peptides are generated by MT1 MMP, the second enzyme, and they precipitate locally and assemble into fibrils. However, in the AAV treated animals, you see that there is a significant reduction. So there is much less deposit of amyloidogenic fibrils, which means that to some extent it has a protective effect in this preclinical model. This is a physiological experiment showing that muscle contractility also improves. Because you can imagine that when these amyloid fibrils are surrounding the muscle fibers, it's going to make contraction much more difficult. Final question, what is the protective effect of the nanobody? Why does number 11 protect against furin and the others don't? The answer is here. For the structural biologist, on the top, you have the crystal structure of the domain where the mutation occurs. This is the mutant. You can see this readily because there is a green sphere missing. That's calcium. I mentioned that the mutation makes it impossible for this domain to bind calcium. Whereas in the wild type, you can see the green sphere, and at the bottom, you see nanobody 11. If you look very carefully, you will see that there is no difference. It's exactly the same crystal structure. And so the conclusion is that the nanobody forces the mutant protein to adopt wild-type conformation. That's basically how we think that it works. Yeah, I'm going to skip this. Other mutations have been identified and the nanobody still has a protective effect. And this is real because, I received an email 4 years ago of a patient who said, I have this disease. I cannot go to the clinic. There is no therapy whatsoever. Can you please enlist me in a clinical trial? Other diseases linked to protein misfolding. Let's do a final experiment. Completely different setting. We're talking about lipid nanoparticles. We all know what lipid nanoparticles are. We have been treated with them several times during the pandemic. Can we make them a little bit more specific? Can we, in other words, put an antibody on the outside, endowing the lipid nanoparticle with targeting properties so that it becomes more specific and that it locally delivers a drug, a vaccine, whatever. The lipid nanoparticles during the pandemic, they were naked. There's nothing on the outside. You just have a set of lipids. And in the lipid nanoparticle, you have the messenger RNA, which encodes spike protein, receptor binding domain, whatever. And then this is going to trigger your immune response. But this is just a naked lipid nanoparticle. So we asked the question, wouldn't it be a good idea to put a nanobody on the outside of a lipid nanoparticle, directed, of course, against a receptor, an antigen. However, there is one important consideration, at least one, and that is that coupling the nanobody to a lipid ideally should be done in a site-specific manner, not random labeling of the antibody because it's going to be destroyed or the biological activity is going to be reduced. We thought of a strategy to site-specifically label any nanobody. And this is done in the following way. I'll recapitulate very brief for the sake of time. What we do is we insert site-specifically a non-conventional amino acid. It looks like a phenylalanine this year, but it carries an azido group, which is highly reactive. And for that reason, you call it azidophenylalanine. It is possible, without going into details, it is possible to site-specifically insert it at any given site in a cDNA. Also a nanobody. And when you have an azido on the one side, and you have a compound with an alkene bond, fluorophore, lipids, a gold nanoparticle, then you can do a very specific click reaction. I'll skip this. It just explains that, you just feed the bacteria whatever they need, and they're going to make a nanobody with an azidophenylalanine at the C terminus. And this is just a way of depicting how the click reaction occurs. It's spontaneous. It doesn't require an enzyme, and it goes extremely fast. By the way, for microscopy, this might also be an interesting approach because you can couple the fluorophore very close to the antigen or to the nanobody. This is, linkage error is a kind of problem, particularly in a super resolution microscopy because there is a certain distance between fluorophore and antigen. You can reduce it using this approach. This work was done by Linglong a while ago from the immunology lab at the veterinary faculty. Let's take a look at how we did this. So we start from a naked lipid nanoparticle. It carries different lipids. One is particularly important because it's a trans-cyclooctane. What we do here is the following, and it looks complex, but it really isn't. It's 2 click reactions, and they are completed within minutes. It goes very, very fast. We first derivatized a nanobody with an azido group, and then we clicked it to the left-hand side. Strain promoted cycloaddition, triple bond. The nanobody is clicked onto this side, but then we still need to click it onto a lipid. And this is done through the right-hand side. This is a tetrazine, and this can covalently couple to a cyclooctane. Double bond, not a triple bond. So 2 reactions, 2 click reactions, and the end result is the VHH is covalently coupled to a lipid that is present in low concentrations in the particle. Then you need to test it. You see that there's a huge difference when you use a naked lipid nanoparticle. There is no engagement with the cells, with the receptor. However, if you equip the lipid nanoparticle with a nanobody, then there is a very strong engagement with cells that express the receptor. Finally, I think this is I'll take this one very briefly. Finally, we also inserted a vaccine within the lipid nanoparticle. It's basically a messenger RNA encoding GFP. You add it to the cells. In the negative control, you see no staining. In the case where you used nanobodies against APN, you see that there is a strong fluorescence within the cells, meaning that the vaccine has been delivered within those cells. And the same basically applied to an in vivo situation where the small intestine was injected in the lumen with these modified lipid nanoparticles. Again, you see a very strong green accumulation within these villi. Of course, it's susceptible to improvement. That goes without saying. Just mention one thing that finally, that in CAR-T, for example, nanobodies might also be of interest because sometimes there is mispairing between VLs and VHs in the CAR-T approach, chimeric antigen receptor T cell, and this causes premature activation of the T cell and so-called T cell exhaustion. If you substitute it with a monovalent hydrophilic binder, in some cases, it might give an improvement. We'll stop here, but I'm just mentioning that you can also go in the direction of plant diseases. Why not? There is one company, Biotalis in Belgium, they developed Evoca, and they're going to use this to treat a certain plant disease. A strange colleague of mine even has the idea of making transgenic shrimp by engineering a nanobody, protective nanobody cDNA into the genome so that it's protected against the Vibrio. It's a great idea, but I don't think there will be many people consuming the transgenic shrimp. So in summary, I try to show that VHHs are a multimodal tool and a very flexible tool for blocking, perturbing, degrading, tracing, crystallizing, ectopically locating proteins of interest. You can make them relatively fast. Within 3 weeks post-immunization, you have the cDNAs. They're economic, versatile, a fast way to generate protein inhibitors. You can scan the surface of proteins, epitope discovery, and so on. Last but not least, I definitely have to acknowledge Wouter, Adriaan, Olivier Gandalf. I mentioned him already during the discussions this morning. He's the guy who makes the libraries, pulls out the nanobodies. And of course, also Linglong, Barth, and Hans. And if time permitting, I'll be happy to take a few questions. Thank you very much.